Hi there. My name is Shaun Mahony. I'm at Penn State. My research group is interested in understanding how transcription factors establish their binding patterns when they are introduced into a new regulatory environment. And today we're going to focus on the Forkhead family of transcription factors, and we're going to show how we've been using neural networks to understand how transcription factors are influenced by chromatin and sequence patterns when introduced into a new chromatin state. So when we think about transcription factors becoming active in a given regulatory environment, we might think of pioneer transcription factors, which have the ability to disrupt that established chromatin environment. And the prototypical pioneer factors are, of course, FOXA transcription factors, which have been shown to bind to nucleosomal DNA, to disrupt that nucleosomal DNA, and to establish competency for the other for binding of other transcription factors during development. However, we also know that FOXA transcription factors can bind to different locations in different cell types. So there's a little bit of a paradox here where there seems to be some degree of cell-specific influence in determining the binding of pioneer factors such as FOXA, and we don't know how much in general they are affected by cell-specific chromatin environments or interactions with other factors versus their innate ability to have pioneering activity. Outside of the FOXA transcription factor family, there's a host of other mammalian Forkhead domain transcription factors. The Forkhead domain itself is expected to have a role to play in enabling pioneering activity for the FOXA transcription factors. We don't know much about these other FOX transcription factors in terms of whether they are also pioneers or how their binding is established. So today we're going to focus on 5 of these factors: FOXA1, FOXC1, FOXL2, FOXG1, and FOXP3. And we're going to compare how they might, how their pioneering activities or their interactions with chromatin environments might change. So the first thing to say about these 5 factors is that they have similar in vitro binding preferences, at least according to what's stored in the ISMB database. They all seem to like to bind this TG-TTT-AC pattern. So we might expect that if we introduce them all into the same chromatin environment, that they might all bind to similar sites. So we're going to test this by overexpressing them in mouse embryonic stem cells, where we know quite a bit about the pre-existing chromatin environment. We're going to express them for 24 hours, and then we're going to measure their binding using ChIP-XO. So when we do this, the first thing you can see is that there are sites that are similarly bound by some of these factors or across these factors, but there are other sites, lots of other sites, where some of the factors bind and the others don't. So there's differential binding. Now, when we examine more globally, the first what we could immediately see is that some of these factors do not bind the same cognate motifs that they will bind in vitro. There's a little bit of variation in terms of what their cognate motifs might be. So when looking at these ChIP-XO plots, we can see that there's 3 main types of motif that these FOX factors bind. FOXA1 and FOXG1 do bind to this motif that they bind in vitro, this TG-TTT-AC pattern. But there's the other factors like to bind or prefer to bind this simpler TG-TTT motif or a tandem repeat of the same type, this TG-TTT, G-TTT, etc. motif. So there does seem to be some variation in terms of what they like to bind in terms of their cognate motif. We would therefore expect that they might bind different sites within mouse embryonic stem cells. When we cluster these sites, these various sites that they bind, we do indeed see that. We see some sites that are preferred by one or another of these factors, like these sites are preferred by FOXA1, these sites by FOXG1, these sites by FOXP3, etc. So immediately we might think that these differences now are determined by their differential cognate motif instances. So we can look at that question by just doing meme motif finding on these various clusters of sites. And to some degree, it's true, at least for FOXA1, again, it has a greater preference to this in vitro bound motif, this TG-TTT-AC motif, and that is enriched at these FOXA1 preferred sites. However, these other clusters of sites don't really seem to have any major motif that is discriminative for their binding. So all of them have this TG-TTT or repetitive TG-TTT motif present. We can dig a little bit deeper using discriminative motif finding approaches, such as the SQLminder platform that we published a few years ago. When we look for k-mers or motifs that are discriminative between these different clusters of sites, we do find some that are discriminative. However, through analyses we're not showing here, we've determined that these various motifs that are discriminative are not sufficiently discriminative to explain why some of these clusters are bound by some transcription factors and not others. There's just not enough presence of those motifs or enrichment of those motifs across a wide enough variety of sites to explain why some factors bind some sites and some bind other sites. So if it's not sequence, then what could be going on at these sites? Well, remember that we have overexpressed all of these factors in the same chromatin environment, but their interactions with that chromatin environment might vary. So we know a lot about the mouse embryonic stem cell chromatin environment, so we can compare where these transcription factors are binding at 24 hours to what was happening in the regulatory environment at those sites in mouse embryonic stem cells. So when we do that, we can see quite a bit of variation amongst these different FOX factors. For example, over half of the sites bound by FOXP3 or FOXG1 are already enriched for these active histone modifications or are already accessible according to TAXIQ in the pre-existing mouse embryonic stem cell chromatin environment. In contrast, FOXC1 and FOXL2, the majority of sites are not pre-marked by active histone modifications or by accessibility. FOXA1 is in between. We can look a little bit more comprehensively using chromatin state analysis from ChromeHMM. Again, sites bound by FOXG1 or FOXP3 are much more likely to be associated with sites that were already displayed the patterns associated with enhancers or transcription start sites in mouse embryonic stem cells. In contrast, FOXL2 and FOXC1 are more associated or have a higher association with quiescent states, which really don't have any of the analyzed histone modifications present. FOXA1, again, is in between. So there's clearly some distinction amongst these different transcription factors in terms of how much they associate with different chromatin states in the pre-existing mouse embryonic stem cell chromatin environment. We also know that there are some sequence differences between these factors. So how do these two things come together to explain differential binding? So that's what we're going to look at and what we have been looking at with convolutional neural networks. So Jianyu Yang in my lab has been building convolutional neural networks that we train with either DNA sequence information or with DNA sequence and pre-existing chromatin information in order to predict the chip exo read counts at these various sites. Now, the reason we're training with DNA sequence versus DNA sequence plus chromatin is we want to see what the marginal improvement in performance is by integrating pre-existing chromatin information into the process. So we can measure that at a very rough level using just the performance of these networks. So the mean squared error on held out test sets. We can see that adding chromatin information into the training process improves our accuracy. It does so relatively more for FOXG1 and FOXP3, which have a higher association with the pre-existing chromatin, active chromatin states, as we saw already. We can look in a bit more detail by looking at individual test set sites in these and the performance of the networks when we train only with sequence or when we train with sequence plus chromatin. Excuse me. When we look at the sites that the networks that are trained with sequence plus chromatin information, we can see that they do a better job in terms of matching the predicted read count versus the true observed read count, especially for FOXG1 and FOXP3. So these clouds have been drawn more along the diagonal from when we introduce chromatin information into the training process. So these networks are more accurate when we're bringing in chromatin information, but does this explain the differential binding that we've observed between these various FOX factors? We can look back at our clustered sites to examine this. So here's our original chip exo clustered data. These are the predictions on these same sites when we use the neural networks that were only trained using sequence information, you can see that this sequence-only trained neural network doesn't do very well in terms of discriminating which sites are bound or preferred by some transcription factors versus others, especially if, say, for FOXG1 here, the sequence-only neural network basically thinks that all of these sites should be bound by FOXG1, even though only some of them or some of these clusters are preferred. However, when we integrate chromatin into the training process, we can see a bit more discrimination. We can see, for example, that FOXG1 is now predicted to prefer these sites that it does in fact prefer, and the same with FOXP3. So what's going on here? What is this information that's coming from the chromatin side of the chromatin sequence and chromatin neural network predictions? We can start examining that with feature attribution approaches. So let's apply some feature attribution approaches, first of all, to the sequence component of these neural networks. So using deep SHAP approaches here, we can look at the sequences underlying these sites and ask if they're positive or negative contributors to the predictions. In general, sequence is a positive contributor. It's a more positive contributor for some sites than others, especially in the FOXA1 network, which is correctly predicting these sites to be bound using sequence features that are preferential at these sites compared to other sites. But sequence information alone isn't enough for the neural network here. You can see again for FOXG1, from the sequence information alone, the neural networks think that all these sites should be bound. And we can go a little deeper into this in terms of what motifs are present. We can use TF-MODISCO, for example, to find what motifs are underlying these sequence attributions. When we do that, we sort of get the same picture that we saw before from just simpler motif finding analyses. FOXA1 definitely likes to find this TG-TTT-AC motif, and the neural networks recognize that. But the other factors, according to TF-MODISCO and the sequence features that the neural network is finding, these are pretty much similar motifs across the board here. They all like to find this TG-TTT motif, and that's reinforcing the fact that sequence is not really discriminating whether these sites are bound or not beyond just the presence of this cognate motif. What is more informative is looking at the chromatin feature attributions. So if we look at deep SHAP but applied to the chromatin features, we start seeing a bit more interesting of a pattern. So for example, here's the attack-seq from mouse embryonic stem cells at these sites that are bound in these various combinations. You can see some sites are accessible before in the pre-existing mouse embryonic stem cells, and some sites are not accessible. And that in itself is a differential contributor to the predictions of these neural networks. You can see that the lack of attack-seq at this set of sites, for example, is quite a strong negative predictor of binding for FOXG1 or for FOXP3. So in other words, the sequence information can be similar across sites that are or are not bound, but that those sequence informations can be counteracted by a negative chromatin feature or chromatin attribution or chromatin feature. So this is for attack-seq. We can do the same with, say, H3K4 monomethyl. The same type of picture is present there, that the relative lack of H3K4 monomethyl at some sites is a negative feature. The relative enrichment is a positive feature for some other factors like FOXP3. And together, they're helping to shape the predictions of these neural networks and potentially informing how these factors are interacting with sequence versus chromatin features. So just as one example here, I wanted to focus in on one particular transcription factor binding site. This is a site that's bound by FOXL2, but not bound by the other transcription factors. So you can see here's one view of the ChIP-EXO data here, which shows that it's bound by this particular factor. The neural network predicts that this should be bound by FOXL2, and it's doing so according to the sequencing chromatin attributions. It's doing so mostly based on the sequence attribution here, on the sequence contribution here. So you can see there's several instances of this TG-TTT motif within this site that are positively contributing to the binding prediction. The lack of attack-seq and K4 monomethyl in mouse embryonic stem cells is not a major issue for the neural network for FOXL2. And it's not like negatively contributing too much to this prediction. FOXA1 does not bind this site. but it's really not affected by this lack of ataxic or K4 monomethyl either. It's really more down to the fact that there's not the preferred motif instance at these sites, the TGTTT AC motif that FOXA1 likes to bind. So that's why the neural network is not making a strong prediction of binding at this site for FOXA1. But in contrast, for FOXG1, which again does not bind to this site, there are lots of sequence features that are telling the FOXG1 neural network to positively predict this as a binding site. But those sequence features are being counterweighted by negative chromatin features. In other words, the lack of ataxic and K4 monomethyl are negative contributors here. So in conclusion, in summary, we think that paralogous transcription factors, this is another example of where paralogous transcription factors might be able to diversify their binding targets based on differential abilities to engage different chromatin environments in pre-existing cell types. And we think that neural networks are a great tool to allow us to start putting apart the various sequence and chromatin predictors of transcription factor binding. I want to thank everyone involved in this work. This was a collaboration with Frank Pugh's lab and Minora Ko's lab. And this is, we're grateful to our funders from NSF and NIH. Thank you so much.